Okay. Good evening, everyone. Today we'll be starting with the week 6 content. I am Seemanti Aditya, a Ph.D. scholar in the Department of Biotechnology at IIT Madras. Today we will be talking about the different experimental techniques where we use antibodies and like how do we use the concept of immunotechniques in the lab, basically. So today we'll be discussing the concepts and also solve some questions at the end. And any doubts regarding the previous classes or like this class as well, we can discuss in the end. So let's begin. So what are immunotechniques? So any technique that uses antibodies to carry out our experiments in the lab is known as immunotechniques. For example, immunodiffusion, immunofluorescence, ELISA, immunoblotting or Western blotting, as we say, flow cytometry, etc. So all of these techniques are routinely performed in most of the research labs and most of the wet labs. So we'll be discussing each of these techniques in brief details, and we'll see how antibodies are used in different in different experiments, and we can make use of this simple concept of antigen antibody interaction and carry out our experiments. So for carrying out the studies, antibody needs to be first purified from the serum of any animal where the antigen is injected, and then the purification is done by different chromatographic techniques like affinity chromatography or ion exchange chromatography. I think you all know what do you mean by affinity chromatography or ion exchange chromatography, etc. So basically affinity chromatography involves the one of the techniques of affinity chromatography is this protein A based purification, protein A based purification of IgG antibodies. So here what we do is we have a column, and in this column, as I discussed in last class or some in the earlier classes, that we have beads. So in each of these beads we have coated with protein A. Now protein A is an shows antigenic property. It has like in case of staphylococcus bacteria. So protein A is an it acts as an antigen. So we use this antigenic property of protein A to coat our beads, and our beads will have protein A on top of them. So now this protein A can bind to the Fc region of our antibody. So Fc region of our antibody is the constant part of our antibody. So this constant part of this antibody gets attached to the beads containing coated protein A, and from there these antibodies can be purified. So basically all these antibodies is trapped over here, and then we are using different buffers and different elution pH, etc., to elute our antibody. So similarly, different techniques. Another technique is an ion exchange chromatography. So here we use beads which are coated with some cation or anion, and then we can pass our antibody or pass our different proteins which will have some ionic charge on them, and we can purify our protein of interest. So very first need for any kind of immunotechnique is to purify our antibodies. So antibodies needs to be purified, and now first how do we generate, or how do we inside an animal, how do we uh, allow the production of antibody? So first, like whatever antigen we are working with, we need to inject the antigen. So this is our antigen, say. So here we'll be discussing how can we purify or isolate monoclonal and polyclonal antibodies. So, okay, before that, I'll just explain in brief what do you mean by monoclonal and what do you mean by polyclonal. So, monoclonal antibody is any antibody which will recognize just one of the epitopes. So, we know no, that the antigen has a binding site to the antibody. So, this is an antigen, suppose, and it can have different kind of epitopes. Okay, so epitope is a region which can bind to the antibody, region that binds to antibody, right? So one particular antigen, so an entire antigen can have several different epitopes. Similarly, similarly, the different antibodies region that will interact with the epitope is called as paratope. So region of antibody that interacts with epitope of this antigen. So basically epitope paratope interaction takes place. Okay. Now we have an antigen. So here you see, look at this antigen property. It has 4 different epitopes, number 1, 2, 3 and 4. So all these epitopes have different kind of binding affinities to different antibody. Now any antibody that can bind to just a single epitope, say an antibody against epitope 1 will be called as a monoclonal antibody. So monoclonal antibody is any antibody that interacts with a single epitope. Okay. Now a collection of monoclonal antibodies, say here if so this is one antigen, it can have 4 different epitopes. Now when you are injecting this antigen into a rat or into a mouse or into a rabbit or horse or any animal and collecting the serum, so antibodies are present in the serum. In the very first class I had discussed that where do we get the antibodies. So we get it in the serum, and that is the serum. What do you mean by serum? Serum is the blood component where you are removing all the cells and also the clotting factors, and whatever you get it just the fluid part or the liquid part of the blood without the clotting factors and the cells, the WBCs or RBCs or platelets, that is known as serum. So this serum will contain, when you are injecting an antigen, it will contain several antibodies against the different epitopes. Say epitope 1 will have antibody number 1, epitope 2 will have antibody number 2, epitope 3 will have an antibody number 3, and epitope 4 will have antibody number 4. So this particular serum that you will isolate, or this particular collection of monoclonal antibodies, is called as polyclonal antibodies. So what is polyclonal antibodies? So polyclonal antibodies is a collection of monoclonal antibodies, right? So so generally, whenever an antigen of our interest, say any protein you are working with, BSA or any kind of antigenic protein, any viral protein, if you are injecting in this animal, and isolating the serum component and isolating the antibodies, you are going to get polyclonal antibody. So most of our work, if we are doing, we can do it with polyclonal antibodies. But in some cases where we require very specific antibody against a specific epitope, then we will require the purification of a monoclonal antibody. So the purification of monoclonal antibody requires the formation of a cell which is known as hybridoma cells. Now here we can here I have to explain a bit. So say this particular mouse, after injecting the antigen, we have to isolate the spleens, because here you are getting the secondary immune response, and here you will get a lot of antibodies will be present against the antigen. You remember, spleen is a secondary immune, secondary lymphoid organ where the antigen interaction, antigen antibody interaction will take place. So it's a place where you can isolate a lot of antibodies. So you are isolating spleen cells, and from there you are getting antibody number 1, 2, 3, and 4 against different antigens. So this is again a collection of polyclonal antibodies. So from spleen, these antibodies, along with these antibodies, you will also have plasma cells. So basically B cells that are secreting these antibodies. So each of these plasma cells are against producing antibody against a particular epitope, say 1, 2, 3, and 4. Now you will fuse the plasma cell with the myeloma cells. Why are we doing this? So we basically want the continuous production of antibodies or monoclonal antibodies. So the property of plasma cell is to secrete antibody, and the property of myeloma cells is to proliferate uncontrollably. Okay. So if we are fusing the plasma cell and myeloma cells, we are going to get a hybrid cell containing the properties of both B cell and the cancer cell. So this particular B cell, which is fused with the cancer cell, will now secrete antibodies continuously, because it will show the cancerous property as well, where it will continuously multiply and it will secrete antibodies. But we need to select our particular hybrid cell, because when we are fusing the plasma cell and myeloma cells, there could be 3 populations that can be obtained. So plasma cell and myeloma cells, if you are fusing, you can get 3 populations. That is plasma cell plus plasma cell, or you can get myeloma cell plus myeloma cell, or you can get a plasma cell plus myeloma cell. So this is the hybridoma. Right. So we need to select this particular fraction of cells. So in order to select this, we use a technique called as, we use a medium which is known as the hat medium. Here we use hypoxanthine aminopterin and thymidine. So what is the concept here? So plasma cell plus plasma cell, if it is fused together, it will not grow for a long period of time, because these both are normal cells, they are not cancerous. So they will die. So they will die after some time. So these cells will be eliminated after some time. Myeloma cells, the fusion or the hybrid of both myeloma cells will grow uncontrollably, and we need to eliminate this population. How we can eliminate this is, it was found that cancerous cells lack the enzyme known as hypo-HGPRT. So this enzyme is hypoxanthine guanine phosphoribosyl transferase. Right. So this enzyme is HGPRT. It is present in all normal cells. It is present, but it is absent in myeloma cells or cancer cells. It is absent, right? So HGPRT, it is absent. So when we are providing hypoxanthine and thymidine, myeloma cells will not be able to utilize this hypoxanthine and thymidine, because it is lacking the HGPRT enzyme, and so it will not be able to synthesize DNA. So HGPRT is responsible for DNA synthesis through the salvage pathway, where it uses hypoxanthine and thymidine, and so myeloma cell, when you are placing in this hat medium containing hypoxanthine, thymidine, and immunopterin, what does immunopterin do? Immunopterin blocks the DNA synthesis by the normal pathway. DNA synthesis can take place through 2 different pathways. One is the normal DNA synthesis pathway, and one is salvage pathway. So immunopterin will block the DNA synthesis through normal pathway. And we are already providing hypoxanthine and thymidine in the media. So myeloma cell will not be able to utilize this media, and salvage pathway for DNA synthesis will be blocked in this particular combination of cells. So only the plasma cell and myeloma cell hybridoma will be able to grow. So we will be able to isolate in this hat media only the hybrid cells of plasma cells, and the cancer cell will be growing. Rest all other populations will be eliminated. Now from here we can isolate 1, 2, 3, and 4
different clones, and the antibodies that we will get after purifying each of these cells or each of these clones will be monoclonal antibody against each of the epitope for the antigen. So you are getting 4 different monoclonal antibodies. So the first technique that we will be studying today is the immunoprecipitation technique. What happens in immunoprecipitation? So it's based on the principle that antigen and antibody is going to interact. Now, antigen antibody interacts, but in a particular concentration where both the antigen and antibody is in equal amounts. What will happen that in a solution, in a solution when antigen and antibody is in equal amounts, it's going to cross-link, like here it is shown, say with increasing antibody, say there is a serum and you have a different concentration of antigen in your serum, and you are adding antibodies of different concentration along this particular gradient. So here your antibody is continuously increasing across this x-axis. Now, what will happen is during the different concentration of your antigen, your antibody will be interacting with your antigen, but it will not be able to form this kind of cross linkages. You see, there is a cross linkage that is formed because the concentration of antibody and concentration of antigen in this region is equal. This is known as zone of equivalence. So here antigen and antibody interaction will lead to the cross linkage with 2 or more antigen molecules. So when there is low amount of antigen and low amount of antibody, there will be not enough antigen antibody interaction, because the antigen antibody interaction is non-covalent interaction and it requires some contact with each other. Now, if there is very few amount of antigen, few amount of antibody, it will be very much, there will be so much of, in so much of solution, they will not be able to find each other and they will be scattered in the solution. So the interaction will be very less. So here we will not be seeing any kind of precipitation. Also, in case of higher amount of antibody and lower amount of antigen, there will not be any kind of, so each antibody will be bound to just a single antigen instead of like that one antibody can bind to 2 antigen, like because it has a valency of 2. Normal antibodies, IgG type, can bind to 2 different antigens, 2 different epitopes of antigen. So here when antibody is more, so you can see that one antibody is binding to just one epitope of the antigen. But only when during the equal concentration of antigen and antibody, one antibody can bind to 2 different antigen, and therefore it is forming a cross link. This cross linkage is resulting in the formation of precipitation, and this precipitation is visible to the naked eye by formation of a white colored precipitate or a grayish white colored precipitate. So in the solution, you will see that if the amount of antigen and antibody is equal, then we will see a whitish colored precipitate. But if either antigen or antibody is in excess or in less amount, so precipitate is not formed, because primarily monovalent binding takes place. What do you mean by monovalent binding? That just one antibody is binding to one epitope of the antigen. This is monovalent binding. This is divalent binding. Right. So immunoprecipitation, here there is one picture which shows how the precipitin line is formed in a solid medium. So here an immunoprecipitation can be performed in both solution phase as well as in solid phase. So in solution phase, we will see a precipitate, just normal pellet-like form. But in solid phase, you will see a precipitin line. So this is what is called a precipitin line, the whitish line that you see. So in this picture, what they have depicted is, in the center we have an antigen. This is a viral antigen, and surrounding wells will have different antibodies or the serum of different individuals. So individual A, individual B, individual C, D, E, F. So the serum that has been isolated from different individuals is placed over the surrounding wells. The idea was to find out which of the individuals have antibodies in their serum against this particular viral antigen. So according to the picture, what do you think that which individuals are going to have the antibody against this antigen? Anyone? So antigen antibody interaction in particular amount will help us give a particular precipitin line. So so here what happens is, here you're putting the antibody, here you're putting the antigen. So it will be diffusing towards each other. Antigen is diffusing here radially. Antibodies will be also diffusing radially. So in the just near the well, the concentration of antibody is very high. The concentration of antigen is very high near the well of this antigen. As it diffuses towards each other, it is going to, the concentration is going to reduce, and we will find the precipitin line at the exact position where the concentration of antibody and the concentration of antigen is equivalent or equal. So the precipitin line here demonstrates the presence of an antibody in the serum. So individual A, individual F, E, D, C. Individual A, F, E, and C, you are seeing a precipitin line, right? So the precipitin line indicates that these individuals have antibodies with respect to this antigen in their serum or in their blood. But you see that individual B or individual D, they do not show any precipitin line. This indicates that individual B and D, they do not have antibody in their blood. Now this technique is known as Octaloni double diffusion. So why it is known as double diffusion? Octaloni was, I think, the scientist. And why it is known as double diffusion? Because in a well, in different wells, we are putting the antigen and antibody, and both the antigen and antibody is diffusing towards each other. So it is known as double diffusion. What is single diffusion? Single diffusion is if we are taking a plate. So here this is an agarose plate, and we are punching wells in this agarose plate, and in this wells we are putting our antigen or our antibody of interest. But in a single diffusion method, what happens is we will have an agarose plate, and in this agarose plate we will pour our antibody or antigen. So our entire plate will have our antigen or antibody, whatever we want, and we will just punch holes in the wells in the plate and pour either an antigen or antibody, whatever we want. So if we are putting antigen in this entire plate, then we'll put antibody in these wells, and if we are pouring antibody in the plate, then we'll put antigen in the wells, and just the antigen or antibody which is present in the wells will be diffusing, but all the in the plate, whatever is present, it will just be present. So this is known as single diffusion, because just either an antigen or antibody that is present in the wells diffuse. But double diffusion means where both the antigen and antibody will diffuse towards each other. So I hope that is clear. So here again, it has been explained that the center well has a viral antigen, while the surrounding wells consist of serum samples from different individuals. So precipitin line is observed in serum samples of individuals that are positive for the antiviral antibodies. So the precipitin line indicates that the individuals have antibodies against that antigen. But individual B and individual D test negative for the antibody, because we do not see any precipitin line over here or over here. We do not see any precipitin line. See, there is no precipitin line. So individual B and D is negative for the antibody against this antigen. So this kind of techniques are routinely used in medical field for detecting the presence of antibody in the blood of individuals against any kind of antigen that they're looking. Next kind of interesting technique that is used is ELISA. ELISA is enzyme-linked immunosorbent assay. Here we make use of enzyme conjugated antigens or antibodies. So we have an antibody, and here we will just conjugate our enzyme. So this enzyme, and this enzyme uses a chromogenic substrate. What is a chromogenic substrate? Chromogenic substrate is anything which is initially colorless, that you will not be able to see any color, but when this enzyme acts upon this substrate, then the product that is formed is colored. So the colorless substrate that is colorless turns colored product when this enzyme acts upon this substrate. Right. So this enzyme could be horseradish peroxidase or alkaline phosphatase, and they have their different kind of substrates. And so ELISA makes use of antibodies that are conjugated with this enzyme. And so any antigen that is interacting with this particular antibody, or so if you are having any antigen that is interacting with this antibody, and so your antibody antigen interaction will take place, and then you will just add the substrate, and finally, you know, this enzyme will be acting on the substrate to show a colored product. So you can measure the antigen antibody interaction by, you know, measuring the OD. So definitely ELISA is used for detection and estimation of either antigen or antibody. So you can either use a conjugated antigen or antibody. Even an antigen which is tagged with an enzyme can be used. So either way, so vice versa, you can just use an antigen or an antibody tagged with an the enzyme to carry out your assays. So ELISA again can be of three types mostly. So how is ELISA performed? Basically, you have ELISA plates, and in ELISA plates, you can coat the wall of this ELISA plate with either antigen or antibody as per your requirement. So first kind of ELISA is indirect ELISA. Here you coat the entire plate with your antigen of interest. So see, if you're looking for patient's serum, whether the patient's serum contains antibody against HIV virus, so you will coat the wall of the ELISA plate with HIV viral antigen. And so you will then wash this particular plate, these wells, so that all the unbound antigens is removed and just the antigens which is bound to the walls will remain. Then you will add specific antibody. So the antibody from the serum of the individuals that you are testing will be added to this well. Then again you will wash to remove any unbound antibody. And so antibodies will be interacting with this antigens, so it will be so it will show an antigen antibody interaction. And we already have an enzyme which is so this antibody is going to interact with this particular antigen. So this is the primary antibody that is present in the serum of the individual or serum of the patient. Now you will add a secondary antibody. So secondary antibody is an antibody that will recognize the primary antibody. So it is mainly binding to the Fc region of the primary antibody. See here, this is the Fc region. The secondary antibody is binding to the Fc region of this primary antibody. This secondary antibody has an enzyme which is conjugated to its Fc region. So the secondary antibody has an enzyme conjugated to its surface. Now, so our primary antibody binds to our antigen. To this primary antibody, our secondary antibody which is conjugated with an enzyme binds to this primary antibody. And finally, when you are adding the substrate and measuring the color, so this is a colorimetric assay, which will give us the amount of antigen which is present in the amount of antibody that is present inside the the amount of antibody that is produced inside the individual or the patient, you will be able to detect that. So it is a quantitative assay. So once you add the substrate, you will see the color is produced, because the enzyme will convert the colorless substrate to a colored product, as you see here in the yellow color. And if you see a higher amount of color, so it will show that higher amount of antibody is interacting with this antigen. That means that the patient's body has an infection, because if it's an HIV virus and it is having a lot of antibodies against the HIV virus, that indicates that the patient is infected with the virus. So higher amount of OD or higher amount of color will indicate a higher amount of antibody in the patient's body. In case of sandwich ELISA, what we do is we coat the well with antibody, in contrast to the antigen that is coated in indirect ELISA. So so the difference between indirect ELISA and direct ELISA is, in indirect ELISA we are using a secondary antibody which is conjugated with an enzyme. In direct ELISA we will use a primary antibody which is conjugated with enzyme. Generally we don't do that because it is quite expensive to generate a primary antibody conjugated with an enzyme. So we usually perform this secondary antibody based or indirect ELISA techniques. Second ELISA technique is the sandwich ELISA, where antibody is conjugated, antibody is coated in the well. Then we are washing it to remove all the unbound antibody. Then we are adding the patient's serum containing antigen. So all the antigen that is present in the serum will be bound to the antibody. Now we will add a secondary antibody that will have an enzyme conjugated with its surface. So you know that one particular antigen can have different epitopes, right? That's why I was mentioning earlier as well. So one antigen is having this epitope, this epitope, this epitope. So one of the epitopes is recognizing the primary antibody that is coated in the well. The secondary antibody is recognize another epitope of the same antigen in another way. So this antibody is coated with, is conjugated with the enzyme. So you see it is looking like a sandwich. So antibody, antigen, and again an antibody. So antibody, antigen, and again an antibody. It can go the other way around as well. Antigen, antibody, and again an antigen. So here this is known as the sandwich ELISA, where again you are adding the substrate and you will measure the color. So more amount of color will show that more amount of antigen is present in the serum of the individual. So whatever you are coating the well plate with, just the corresponding thing you are measuring in the patient's sample or individual sample. So if you are coating the well with antibody, you are measuring the sample, you are measuring the amount of antigen in the blood. If you are coating the well plate with antigen, you are measuring the amount of antibody in the blood. Now, the third type of ELISA is the competitive ELISA. Here, what happens is you are isolating an antigen from the blood. So you are isolating antigen from the patient's sample, and you are first incubating this antigen with this primary antibody. You're already incubating it. So what will happen? The antibody population will be of two types. One will be bound to the antigen and one will be free. So the amount of antigen that is present in the patient sample, if it is high amount of antigen that is present in the patient sample, then more antibodies will be bound in this step itself. So if you are pre-incubating the antibody with the antigen, more amount of antibody is already bound to the antigen. So the amount of free antibody that will be present will be less. So this free antibody, so this particular mixture of antibodies containing both bound antigen as well as unbound antigen will be added to the well plate, where again we have coated it to an where antigen is already coated in this well plate. So this mixture of population of antibodies, unbound and bound, antigen bound and unbound antibodies will be added to this well. Now the amount of free antibodies that are present, all the free antibodies will be bound to this antigen that are coated in the well. After washing and after adding the substrate and measuring the color, you will be seeing that whatever this will demonstrate, the OD of this will show if the OD is higher. What will that indicate? So if basically the OD is higher, means that the amount of free antibodies is more. That means the amount of antigen in the patient sample is less. So OD high means antibody, antigen is less in the patient sample. But if OD is less, what do you mean by if OD is less? That means the number of free antibodies that were present is less. So if the number of free antibodies that are present is less, that means already the antibodies were bound with the antigen present in the patient sample. So the antigen in the patient sample is more. So competitive ELISA, the technique for analysis is just opposite to what we had observed in indirect and sandwich. So in indirect and sandwich, there is direct correlation between the measurement of color and the concentration of antigen or antibody. So if higher OD means higher antigen and antibody, lower OD means lesser amount of antigen or antibody. But in case of competitive ELISA, just the opposite. If OD is higher, the amount of antigen or antibody is less. If the OD is lower, then the antigen is more. Hope that is clear. Next technique that we will go through was immunoblotting or Western blotting. So one of the problems in ELISA is we cannot determine what is the molecular weight of the protein that we are looking about, or what is the molecular weight of the antigen that we are looking. So Western blot is a confirmatory test that whatever protein you are looking for or whatever antigen you are looking for is exactly the same antigen, because it will have a particular molecular weight, and that molecular weight will denote that, okay, this is your antigen of interest or this is your protein of interest. So this is a confirmatory test. In any kind of experimental labs, you'll see that Western blotting is done for detection of protein expression. And this is kind of a confirmatory test for determining the presence or absence of the protein. So because you can easily determine the presence or absence of protein with the help of the molecular weight marker of this protein or antigen. So it is similar to ELISA. Western blotting is similar to ELISA, but here protein or antigen is in the denatured form, because in Western blot what we do is we have a mixture of proteins that we have from the cell or we have from the tissue, and we just denature it in presence of SDS and beta markaptoethanol. So we denature it in order to give a particular shape, just a linear shape to the protein. So this linear shape will destroy all the conformational epitopes. So what are conformational epitopes? So this is a protein, and in denatured state it is becoming this. So if there is an epitope which is present over here, this will be lost in the linear shape. So denaturation of protein will be will lead to loss of conformational epitopes. So in Western blotting, we cannot recognize proteins which are denatured. So for that we need to do ELISA. But most of the epitopes or most of the antibodies that are designed for the proteins in commercial cases, they are designed against the primary sequence of the protein. So that is not an issue in the normal commercial cases or in the experimental labs. But if you are looking for some other newer conformational epitopes, antibodies against this epitope, so then for that you need to perform an ELISA test. So yeah, so a protein antigen is denatured with the help of SDS. So SDS denatures protein, and three-dimensional structure of protein is lost. So we do not recognize conformational epitopes with antibodies in Western blot. Also, another difference between ELISA and Western blot is we use chromogenic substrate for both Western blot and ELISA. But here the substrate that we use should be different from that of ELISA, because in ELISA we need to have a product that is water soluble, because we need to measure the OD. So in measuring the OD, we cannot have a precipitate, because that will not give a good that is not going to be a good technique to determine spectrophotometrically. So water soluble product is present, like when the substrate is used, after it is changed to the colored product, the colored product is water soluble, so we will be able to measure the OD in ELISA. But in case of Western blot, we do not require to measure OD. We need to see it through our naked eyes whether the precipitate is formed or not, whether the product is formed or not. So the product needs to be water insoluble. So water insoluble product will show a band in the gel or in the blot. So let us see how the Western blot goes. So basically you have a mixture of protein antigens. These protein antigens are denatured in SDS in order to give them a primary shape. Why it is denatured? Why denaturation is important in SDS? Because in SDS we are trying to separate the proteins or resolve the proteins on the basis of its molecular weight and not on the basis of its shape. So all the protein needs to have a similar shape, that is the primary sequence of the protein. So we need to denature it in SDS. So this SDS treated protein mixture is added to a well. This is known as a Polyacrylamide Gel Electrophoresis. This is a polyacrylamide gel. For protein, we use a polyacrylamide gel. For DNA, we use agarose gel. So polyacrylamide gels are having lesser pore size because proteins are small molecules. So yeah, so the mixture of proteins is added to this well, each of this well in this polyacrylamide gel. And this will be moving towards the direction from negative to positive. SDS, apart from denaturing the protein, it is also adding, this is a detergent, so it is adding uniform negative charge to all the proteins. So uniform negative charge. So all the proteins will be migrating from the direction of negative to positive, because these are negatively charged, so it is going to move towards the positive charge. Now all the proteins which are larger proteins, so this will stay in the top part of the top portion of the gel, smaller proteins will be migrating towards will be resolved towards the lower end of the gel. So this is a vertical gel as compared to the horizontal gel that we use in in DNA. We use a horizontal gel. The gel is run in this way, but in protein or Western blotting, the gel is run vertically like this, because there is a influence of gravity as well in order to resolve the proteins properly. So, after dissolving this protein from higher molecular weight to lower molecular weight, we need to transfer this dissolved protein into a membrane or into a paper. It's like a paper kind of thing. So this membrane is known as PVDF membrane. We can also use nitrocellulose membrane. So all these membranes are used for transferring these proteins after running the gel. So gel is run in order to dissolve the protein into its particular molecular weight. Now we cannot see the proteins over here, so we need to perform other experiments like this, like the Western blot, or incubate it with the antibody of interest. So we need to transfer it to a membrane, because if we do that in a gel, the gel is very fragile. It's going to melt or it's going to break. So we cannot perform the further experiments. So the gel is used just to dissolve the proteins into its particular molecular weight areas. And here, as you are seeing the proteins, as they have shown the proteins, we cannot see the proteins through our naked eyes in the gel. It will be just invisible. So we need to transfer it, hoping that it has got transferred into the membrane. After you have carried out this procedure of transferring it and hoping that all your proteins that were dissolved earlier in the gel has transferred to the membrane, then you will carry out incubation with your enzyme linked antibodies. So this proteins will have a specific antibody that will bind to it, and those antibodies will have enzyme linked to it, similar to Elisa. And so once this antigen is there and this antibody is binding to your antigen, and you're adding, after that you're adding the substrate. So the enzyme that is linked to your antibody will convert the colorless substrate into a colored substrate, and you will see a colored band. So this colored band needs to be insoluble, because if it is soluble in water, you will not be able to see any band over here. So the substrate needs to be modified according to that. So in Western blot, what happens is, I'll just repeat quickly. You are taking a mixture of proteins, dissolving it in a polyacrylamide gel, transferring that all the proteins that are dissolved in the gel to a membrane, and then this membrane you are treating it with antibodies that are conjugated with enzyme of your interest, with your enzyme that is going to act on a chromogenic substrate. Then you're incubating the blot with this is called a blot. This particular membrane, or after transferring, is called a blot. And so you will just incubate your blot with your substrate, and then finally you will be able to see a band. So this band will indicate that your protein of interest is present. So this is a confirmatory test. Also, how to indicate the molecular weight of this protein, you're running a subsequent ladder. So this ladder is a predefined molecular weight ladder, which is available commercially. So if you are in this gel, so one well you are running your protein, the other well you are running the ladder. So your ladder you will be able to see through your naked eyes. So each of the colored bands, so these are colored ladders. So you will be able to see a green band, red band, blue band. So all of these colored bands will indicate proteins of a particular molecular weight. Say this is 10 kDa band. This is a 9 kDa band. This is 1 kDa band. So like this, you will see a marker ladder can be run adjacent to your protein of interest. And if your protein of interest falls between, say, 10 kDa and 9 kDa band, so your protein basically lies in between this molecular weight range. And if you find out in your literature that your protein is actually of that particular molecular weight, so you get the confirmatory test that your protein is present in your sample. This is how Western blot is performed. Another kind of technique that is used is immunofluorescence. So immunofluorescence is also used for determining where exactly your protein is localized inside the cell. So all of these techniques like ELISA or Western blot, you do not know exactly in your cell, in your live cell, where is your protein localized. So here what you do is you're using an antibody against the protein. This antibody is labeled with a fluorophore. So this is a image of a dendritic cell. You see red colored dendritic cell, and this is stained with, the nucleus is stained with DAPI. DAPI is a stain that stains the nucleus, and all this green color fluorescence you see over here, this green fluorescence is actually coming from an antibody that is interacting with the protein beta-actin. Okay, so beta-actin, against beta-actin, one antibody that also is tagged with, the antibody is also tagged with a fluorophore. So this image is basically of dendritic cells that are attacked with Listeria monocytogenes, and all these Listeria monocytogenes will have beta-actin inside their body, and this beta-actin is acted upon by, is incubated with anti-beta-actin antibody, which is conjugated with fluorophore, and so we will be able to see a green colored fluorescence. So this red colored fluorescence is the inherent property. This is a genetically engineered Listeria monocytogen, so it will show red colored fluorescence. The naturally occurring Listeria will not show this red fluorescence individually, but yeah, this is genetically engineered in order to visualize it more clearly. But the green color fluorescence part that you are seeing over here is actually the antibody that is detecting the beta-actin, and because it is conjugated with a fluorophore, if we are seeing it in the fluorescence microscope, what does fluorescence microscope do? So the fluorescence microscope, in the presence of green colored, like blue colored excitation, you will be seeing the green colored emitted light, and only if in the presence of this protein, if it is present, if your beta-actin is present in the cell, then only the antibody will bind to the beta-actin, and when we are exciting it with blue light, we will be able to see emission of green light, or the green colored beta-actin regions. So this is another utilization of immunotechniques. And the last technique that we'll see is flow cytometry, or flow FACS. FACS stands for, this is the machine's name. This is actually fluorescence activated cell sorter, and it's a very useful machine actually, because it has a several function. So on the basis of the name, what do you feel? Like the fluorescence activated cell sorter. So this means that whatever fluorescently labeled cells, or whatever cells that are showing fluorescence, they will be sorted on the basis of their different criteria or characteristics. Now in flow cytometry, we have three important components, that is optics, electronics, and fluidics. Fluidics. So these are the different components of this machine. So this is entirely a capillary tube. So in this capillary tube, you are putting the sample. So the sample is present in a, say, kind of tube, fax tube, or an Eppendorf tube. Your sample is present, and this sample you are loading it in a capillary tube. So all this is a very thin capillary tube, and will have space for just a small amount of cells to go. So this capillary tube is filled with a fluid which is known as sheath fluid. Sheath fluid is used to maintain the buffer of the system. Like the cells has to be maintained in a particular buffer, because otherwise alteration in pH will lead to cell death. Cell will burst basically. So it has to be maintained in a buffered condition. So this capillary tube is maintained in a buffered condition, which is having sheath fluid. And so samples containing stained cell is present in this, is presented to this capillary tube. And once you're putting it inside the machine, all the cells will be, taken up by this capillary tube, by this capillary action. So this particular term is hydrodynamic focusing, which means that in this particular capillary tube, the cells from the suspension will be moving in a single line, that is, or a single file. It will have only one line of cells, so that we can just identify a single cell from a population of cells. So through this capillary tube, a single cell is basically passing. So as the focusing is going on, so this part of the fax machine is called as the fluidics, because all this fluid part of the cell or the fluid part of the sample is loaded. So once the cells are passed through this thin capillary tube, there will be a region where there is no capillary tube and just the cells are passing as a single stream of water or a single stream of buffer coming. And so from here, you will get a laser light source, and the light source will be falling on a single cell, just one cell. And this light source is known as the optics, where we will be able to see that all these laser lights that are used for determining different kind of fluorescence. So we need different lasers or different excitation for different kind of colors: red, blue, green, yellow. There are different colored samples that can be present, and different, so we need different laser light source. So laser light source comes under the component of optics inside the flow cytometer. So laser light source will pass light through your cell. So there will be, once the light is passed through your cell, there could be different kind of scattering. One is the scattering which takes place because of the size of the cell. So this is the cell, light is passing through your cell. Okay, so because of the size of the cell, there is one kind of scattering that is known as the forward scatter. And another kind of scattering takes place because of the light that passes through the cell. It will be refracted in different places. So this scatter is known as the side scatter. So any cells basically will be differentiated on the basis of their size and granularity with this presence of forward scatter and side scatter. So more the amount of forward scatter, more big is the cell. Less the amount of forward scatter, smaller is the cell. So forward scatter, if it is higher, cell is big cell. If side scatter is higher, granular cell. What do you mean by granular cell? So granular cell is anything that has a lot of granules inside it. So yeah, any kind of lot of mitochondria, lot of organelles, vesicles, this, that. So those are granular cells. Granulocytes, basically the as we know, granulocytes are some cells which are having a lot of granules inside it. So that is okay. So that is one part of facts. So where we'll be able to differentiate on the basis of size and the granularity. Another part is where you are differentiating the cells on the basis of fluorescence. So fluorescence cells can be differentiated based on the light source. So if you are having different laser lights which are recognizing your stained samples of yellow, blue, or green color. So if you are having, say, green GFP tagged cell, you are having RFP tagged cells. What is GFP, RFP? GFP means green fluorescent protein, RFP means red fluorescent protein, YFP means yellow fluorescent protein, and etc. So all of these fluorescent proteins will be excited through different filters, excited through different wavelengths, and we will be seeing the emission in different wavelengths as well. So here we can differentiate between the different colored samples. So facts can be used for differentiating the size and shape as well as the granularity of the cell. It can also be used to differentiate between the surface properties of the cell, because you are having certain protein which is tagged with GFP, tagged with RFP or YFP. That also you can differentiate in facts. You can also differentiate, sort each of these cells into each of these categories. So the part where you are, just combining this optics and fluidics, like where you are combining this graph of optics and fluidics, that is known as electronics. So electronics will show us the basically all these compilation of all the fluidics and optics part and give us a graph which will show us different population of cells. So here you see neutrophils, monocytes and lymphocytes. So this is a forward scatter and for versus side scatter graph. So different sizes, cells are of different sizes. Neutrophils are larger than neutrophils are more granular than monocytes, so it will show a greater side scatter. Lymphocytes are the least granular size, so it will show less side scatter. Also, on the basis of the forward scatter, you can differentiate the size of the cell. So lymphocytes is smaller in size as compared to monocytes or neutrophils, so it is accordingly placed in this graph. Also here you can differentiate between CD4 and CD8 cells, so we have to tag it with different kinds of fluorophores or different kinds of different kind of fluorophore. So CD4, say you're tagging with red color, CD8 you're tagging with green color, so you can easily differentiate between the cells expressing CD4 receptors and CD8 receptors. So in a population of cell, you can differentiate between larger cells, smaller cells, and medium sized cells, as well as granularity. So this is the forward scatter graph. This is the side scatter graph. So on this axis, higher it will if the cells are more towards this axis, this is the biggest cell, and on this axis, this is the most, say, granular cell. This axis will show most granular cell. So you can easily figure out from this graph whatever is your requirement, and you can just select that graph, that portion of the graph, and sort your cells accordingly. So you will just get one. So if you're having a proper mixture of cells, you're having a mixture of CD4 and CD8 plus cells, and you want to separate it, so you can collect it in different tubes. So you will be needing different laser lights, and once the laser light recognizes a red cell, it will sort it into a different tube. Once it recognizes a green cell, it will sort it into a different tube. So we are using the term as sorter. It is also used for sorting the different cells. So this is all about facts. So I hope you understood. If any questions, you can ask me now. Then we'll move on to the assignment. Some questions, I'll just move on.
Ma'am,
Yes.
assignment fifth question, ma'am.
What?
Fifth question, ma'am.
Assignment 5, fifth question.
Yeah.
Okay, let me check. Okay. So the question is, which of the following non-classical MHC molecule is required for the catalysis exchange of clip peptide with
Ma'am,
antigen?
no, ma'am. Sorry, ma'am. Sixth question, ma'am.
Six.
HCA, HLA something question, ma'am. Sixth question.
Okay. So if 345 numbers of DRA and
Yeah.
45 numbers of DRB genes are present in human, how many types of HLA class 2 could be expected? Right. So this could be this has to be multiplied, right? So 345 numbers of DRA, 45 numbers of DRB. So you have to multiply this particular genes in order to get the recombination of HLA. So these are HLA type of molecules. So 345 into 45, what will be the what is 345 into 45 comes? Let me see. Yeah, so 345 into 45 is option number C, 15,525. So yeah, so basically this question is like how many possible recombinations you can get of different genes. So I had discussed like how you can, how T cell receptor, B cell receptors are there, how they are managing to get so many kinds of interaction, so many kinds of sequences in them. So we were discussing that how the diversity is maintained. So also like different techniques are used, different kinds of ways that T cell receptor and B cell receptor use for increasing the diversity. So one of there I had mentioned about that how the diversity was maintained. I don't know, in week 3 or week 4. Let me just check through the slides. Yeah, so I'll just share the screen. See, so we were seeing how many types of diversity can be maintained. So just you have to multiply all the segments or all this number of whatever segments or genes that are present. So 41 into 23 into 6 will give 5,658. So similarly, the question is like this type of gene is, say, gene A is this many number, gene B is this many number. So if you are recombining it, how many numbers will be present? So you just have to multiply it and the answer will be that. So this question, similar to the concept which is used here, the possible number of combinations, you have to just multiply the number of genes. Did you get it?
Get it, ma'am.
Yeah. Okay. Shashwata has asked, can one antibody bind different epitope of antigens? No. One monoclonal antibody will not be able to bind to different type of epitopes. One monoclonal antibody will not be able to bind to different epitopes. So if you're using a if you're using a polyclonal antibodies against one antigen, then it can be binding to different epitopes of the antigen. So one monoclonal antibody can bind to only one epitope of the antigen. Okay.
Okay, ma'am.
Yeah. Now quickly, I'll just go through the questions. So pay attention over here. So also one poll will be shared, I guess. So yeah, what do you think of this question? Correct. The question is pretty simple. The best agglutination or precipitation. What do you mean by agglutination? Agglutination is a term again used for antigen antibody interaction, and where you are getting a precipitate or precipitin line. So if you are getting an agglutination or precipitation in the most region where there is equivalent concentration of antigen and antibody, so the correct answer is answer B. Right. So yeah. So the next question. What does forward light scatter and side light scatter? I just discussed. Forward light scatter and side light scatter. Forward light scatter is basically going to determine this. This is forward light scatter. Side light scatter is this. So what will it be? So yeah, so so forward scatter will demonstrate the size of the cell, because it is going to, the light will pass through the different diameter of the cell. So if a bigger cell is there, the forward light scatter will be more. Smaller cell means it will have a small forward scatter. So it will demonstrate the size. So forward light scatter is for the size of the cell. And side light scatter, it is going to, through the inside of the cell, and will diffract or diffract more if there is more granules inside the cell, or if the cell has lot of vesicles inside it. So the side light scatter is going to denote the granularity. So the correct option is size and granularity of the cell respectively. Okay, answer is A. This third question. So right, the correct answer is option D. Flow cytometry can be used for analysis of cell surface as well as intracellular molecules, because we can tag it with different colors and use different lasers. It can also be used for analyzing the cell size and volume through forward scatter or side scatter. It can also characterize different cell types indicating that CD4 cell type, CD8 cell type. So it's different dendritic cell, monocytes or macrophages. So all of these cells can be characterized using flow cytometry. The correct answer is answer D. So that's right. This is the fourth question. Okay, so for this question, which method is used for detecting the presence of serum antibodies against HIV? ELISA is one of the techniques, correct? That is right. But also we use Western blotting. As remember, I was saying that Western blotting is a confirmatory test. So it's a confirmatory test. Okay, so if you are having if you're taking an ELISA measurement, it might give false positive results or false negative results, because you are not sure that whatever antibody is interacting with your antigen, whether it is your antigen or your antibody. Because we have also experience, we also know about the term called cross reactivity, where an antigen antibody interaction can take place even if they are not they are not the just specific interaction. There could be some non-specific binding as well. So in order to avoid that, also HIV testing is done to Western blotting is done to confirm the HIV. So the answer will be both option A and B, because Western blotting is also an essential test to confirm the presence of antibodies against HIV. Not just ELISA will be a conclusive test. So the correct answer is option number C. Right. And this will be the last question for today. So what could be the answer for this? So what happens in a Western blot actually? You're running the gel. Gel is having mixture of proteins, it is dissolved. Then you're transferring it into a blot. So here you're expecting that all of your proteins are, transferred. So then you are incubating it with your antibody of interest. And then you will be incubating your secondary antibody. This is your primary antibody incubation. Then you will be incubating with secondary antibody. Okay, so before that, what is blocking? What is blocking? So any kind of, like when you are using a Western blot, so wherever your proteins are present, or it is resolved, after you're transferring it into your blot, before incubation with primary antibody, you need to give a blocking step. So here what happens is you are incubating it with BSA. So all the regions where there is no protein band over here, it will be bound by BSA. So that this is used just to avoid non-specific binding of antibody with your antigen, because there might be some antigen antibody non-specific binding as well. So just so your protein of interest is present over here, the band is present over here. So all the remaining empty region is bound by BSA. So this is known as blocking. And so blocking is an essential step in order to avoid non-specific interaction. So if your blocking is not there, then your there might be some non-specific binding. So yeah, so these are the essential steps. The transfer is an essential step, blocking is an essential step, proper concentration of primary antibody to bind to your antigen is an essential step. Also secondary antibody incubation is an essential step. So if no signal is observed in your Western blot after performing the Western blot, one of the reasons could be that it has not been transferred properly in the blot. Okay. Also, it could be the primary or secondary antibody is very diluted, so you're not seeing any kind of like any kind of color or any kind of band. Also, there is not insufficient incubation with detection reagent. So basically all this particular three criteria that you are using, if it is not present, like what is detection reagent? Detection reagent is the substrate that you are using for generating the color. So if the substrate is not proper, or if it is not properly incubated, you will not be seeing any band. If it is not transferred only, you will not see any band, because it is not transferred. All the gel, it is present in the gel itself. All the protein is present in the gel itself. And also, if you're using primary and secondary antibody in a very dilute concentration, the secondary antibody will not have enough enzyme to convert the substrate, so you will not see any band. So all of these reasons will lead to no band. This is not essential for not observing any kind of band, because if blocking is insufficient, then also you are going to see some non-specific band. No signal means there is no band at all. Insufficient blocking may lead to non-specific bands, but not no bands. Okay, non-specific bands might be observed. So yeah, so the correct answer is option number B, 1, 2, and 3. So no signal will be observed only in these regions. So yeah, that's all for today. I took a little longer time. Sorry for that. I'll just go through the chat. Anybody has any questions other than this? Okay, so if nobody has any other questions, we can call it a day and we can just meet in the next Friday with new topics. Thank you everyone. Have a nice weekend. See you again on Friday 7 p.m.